Hi guys, I am your biology teacher Dr. Mahmood. Today we will continue with the same topic in part 4 where we left in part 3. Today we will study the structural classification of proteins. I will explain you the main difference between fibrous and globular proteins. We will also study the importance of amino acid sequence in the structure of proteins, and we will also touch very briefly the structure of prions, which are the self-reactive proteins of proteins. Based on their structure, proteins are divided into two main groups. These include fibrous proteins and globular proteins. Difference in their structure is based on the arrangement of the polar and non-polar R groups in a polypeptide chain. Fibrous proteins have no peculiar arrangement of their polar and non-polar R groups, whereas globular proteins curl up in such a way that all of the hydrophilic R groups are arranged outside, while hydrophobic R groups are arranged inside towards the interior of the protein. These proteins play a structural role in the body. For example, collagen forms tendons, cartilage, bones, teeth, and the walls of the blood vessel. They are long, narrow, and thread-like in appearance. For example, silk and keratin. They are insoluble in water. They have repetitive amino acid arrangement. For example, every third amino acid in the collagen is a glycine. The polypeptides which make up fibrous proteins, for example, collagen molecules, are in the shape of stretched out helices. Globular proteins play a functional role in the body. For example, transport proteins, enzymes, hormones, etc. They consist of coiled and folded polypeptide chains forming spherical shape, for example, hemoglobin. They are soluble in water. They are highly sensitive to changes in the temperature and pH. Single chain proteins have tertiary and multi-chain proteins have quaternary structures. These proteins perform metabolic functions, for example, mitochondrial membrane proteins. The primary structure of proteins is a conserved sequence of amino acids. It is determined by the sequence of genes in DNA. Even a slight change in the amino acid sequence can affect protein's ability to perform its function. This is just like changing a single letter can drastically affect the meaning of a word, tasty versus nasty. For instance, the substitution of one amino acid for another at a particular position in hemoglobin, the blood protein that carries oxygen, causes sickle cell disease, which is an inherited blood disorder. Even though 145 out of the 146 amino acid in hemoglobin are correct, that one change is enough to cause the protein to fold in a different shape, which alters its function and in turn causes the disease. Now we come to prions. Prions are misfolded proteins associated with severe brain disorders. Kuru is one such disorder, first described among a tribe in Papua New Guinea in 1950s. This human brain disease was transmitted by cannibalistic ingestion of an infected person's brain, which transfers the prions to a new host. Misfolded versions of abnormal brain proteins infiltrate the brain, converting normally folded proteins into the abnormal shape. Clustering of misfolded proteins eventually disrupts brain function. Here you can see how prion proteins bring about the destruction of brain tissue. Prions in cattle cause mad cow disease. They also cause fatal weight loss in several animals, including deer, elk, and moose. We end up our discussion on proteins, and in the next lecture we will study nucleic acids. I am thankful for listening to my lecture. Bye for now.